Now I will be dealing with shoot apex organization. First I will make the diagram for that and then explain it to you. Yes, so shoot apical meristem, we have to study the organization, different theories put forward by the scientist to explain its structure. Yes. Now this region, yes, this region which I am representing here, this is going to represent shoot apical meristematic zone. This is shoot apical or shoot apex meristematic zone, meristematic region. Now the shoot apical meristems, it will cut the cells on the flanks. Flanks means on the sides. Yes, the shoot apical meristem, it will form certain derivatives on the flanks, on the sides, and these derivatives, they are going to form leaf primordia. Yes, this is what I have shown here. Leaf primordia, as the name suggests, the structure is later going to be converted into the leaf. Along with that, on the flanks, on the sides, there is formation of the structures, that is the axillary bud. Yes, the axillary bud is here. I have shown it here. On the flanks, there is formation of axillary bud. You have studied previously in the morphology, the bud is immature shoot. It is embryonic shoot. So this axillary bud, it can grow, results in the formation of a branch. Yes, it will form the lateral stem, it will form the branch, or it can also be converted into the flower. Now in this shoot apical meristem, in this shoot apex, you can see some of the region is broader, then it is narrow. Again this is broader. The active vegetative meristem, it is going to show this type of the condition. कुछ areas में broad हो जाएगी, फिर narrow हो जाएगी, and this is going to occur again and again. जिन एरियाज के अंदर ये ब्रॉड हो रही है वहां पर ये बनाएगी लीफ प्राइमोडिया एंड अलोंग विद दैट देयर इज फॉर्मेशन ऑफ एक्सिलरी बड and these broaden areas later you can guess what they are going to represent. They are going to represent the node. So this broaden area going to represent the node, and the narrow area between the two nodes is going to represent internode. This is clear? Let's write it down. On the flanks, so you understand flanks towards the sides, it will form leaf primordia and axillary bud. Axillary bud. It will show broadening and narrowing. Yes, it will keep on showing broadening and showing the narrowness. The broaden areas, they are going to represent node. The narrow areas, they are going to represent internode. So as I have discussed here, that this shoot apical meristem form derivatives on the flanks which will be converted into the leaf primordia and axillary bud. Along with that, it will also cut the cells on the lower side. As we have discussed earlier, on the lower side it will cut cells which will form the different tissues of the shoot. So this is journal about the shoot apex. So now we are going to deal with the shoot apex organization which were given by the different scientists, right? So now we will deal with the different theories to explain shoot apex organization. Here I will be dealing with three theories: apical cell theory, right? Then histogen theory, tunica corpus theory. So let's start with the apical cell theory. Apical cell theory, it was given by the two scientists, that is Hofmeister and Nageli. Now according to this theory, the shoot apical meristem is made up of only one cell. Yes. The shoot apical meristem is made up of only one cell, and this single cell activity has the ability to form complete body of plant. Yes, so according to this theory, the shoot apical meristem consists of one cell. This theory is very much applicable for the lower plants which have simple body organization like algae as well as most of the bryophytes and pteridophytes. So this is applicable for algae, most of bryophytes and pteridophytes. But for the higher plants which have complex body organization, there this apical cell theory is not applicable. Instead of that, two theories which were put forward, that is histogen and tunica corpus theory. Now histogen theory, it was given by the scientist Hansteen. According to this theory, the shoot apex is made up of three histogen. It is composed of three layers. Three histogens or three layers. So I am writing the name of these three layers. The outermost layer, that is dermatogen. Right. The innermost layer is plerome and the middle one is peribulum. So three layers: dermatogen, peribulum, plerome. These are the three layers part of the shoot apical meristem according to the histogen theory. So let's see now what are the functions performed by these three layers. Now before dealing with the function, I would like to tell you something about the different tissue system present in the plant or present in the organ. In the organ, there is presence of three types of tissue system: epidermal tissue system, ground tissue system, and vascular tissue system. So here I am writing it down. Three type of tissue system: epidermal, ground and vascular tissue system. Three types of the tissue system associated with the organ. Epidermal tissue system, it is going to constitute, yes, it constitutes outermost layer of any organ. Yes, what is that? Epidermis. So this is going to constitute epidermis. Right? Now coming to the ground tissue. Ground tissue consists of cortex, pericycle and pith. What is this? Cortex, pericycle and pith. This we will discuss in detail. Yes, this we will discuss in detail in the forthcoming lecture. Now the third one, vascular tissue. You have studied in the previous classes. This is going to consist of xylem and phloem. तो सबसे बाहर वाला पार्ट एपिडर्मल टिश्यू सिस्टम, सबसे अंदर वाला पार्ट दैट इज वैस्कुलर टिश्यू सिस्टम, और इन दोनों के बीच वाला पार्ट दैट इज ग्राउंड टिश्यू सिस्टम. एपिडर्मल, ग्राउंड एंड वैस्कुलर टिश्यू सिस्टम. Right? So now with this much of the background, we can deal with these three histogens. Dermatogen. You can simply guess dermatogen. It is the outermost layer. So which tissue it will be forming? Epidermis. It is involved in the formation of epidermis. Yes. Now the plerome we can see here. Plerome is going to form xylem, phloem, vascular tissue. On the outer side, it will form pericycle. On the inner side, it will be forming pith, xylem, phloem, along with that, pericycle and pith. Xylem, phloem, outer side, pericycle, inner side, pith. These three things together going to represent steel. Yes? So these three things together going to represent steel. Yes, then what is left? What is left? Epidermis is done. This xylem, phloem, pericycle, pith is done. Which part is left? The part of the ground tissue that is known as cortex. Yes, so cortex will be formed by peribulum. So this is fine. Now after the histogen, now we can look into the second theory which was put forward again for the plants which are going to have complex body organization. Tunica carpus theory, given by the scientist Schmidt. According to this theory, now the shoot apex, it consists of only two regions. Instead of two regions, histogen is saying how many regions? Three regions, but here only two regions. Two regions. The outer region is tunica, right? The inner one is corpus, right? So let's say this is shoot apical meristem. Let's say this is shoot apical meristem. Then the outer region is tunica. I'm labeling it like this. Outer region is tunica, which is made up of generally single layer of cells. अंदर जितना भी पार्ट है, एक लेयर को छोड़ के बाकी अंदर जितना भी पार्ट है, this is going to represent corpus. Rest is going to be the part of corpus. Got it? Outer layer tunica, and inside everything which is present, this is corpus. Now the cells of the tunica, generally they are small size. Corpus larger size. Right? Now in the cells of tunica, let's see what type of the division is there. I've shown this cell in the transfer section of organ. Yes, this cell is in the transfer section of organ. And if this cell is showing the division like this, ऐसे division हो रही है. What type of division it is? It is perpendicular to the axis of cell. If the division is like this, perpendicular to the axis of cell, this type of division, it is known as anticlinal division. Yes, perpendicular to the axis of cell, that is anticlinal division. Right? This division is shown by the cells of the tunica. Let's see what will happen in corpus. Corpus, this division is present. Anticlinal is present. But along with that, the cells will also show the division here in this plane. Also show the division in this plane. Now what is this plane? Parallel to the axis of a cell. This type of division, it is known as periclinal division. You got this or not? It will show anticlinal as well as periclinal. But please mind it, please remember it, the periclinal divisions, they are more in occurrence as compared to the anticlinal one. Now the outer layer, it will form the epidermis, and the inner layers will form ground tissue and vascular tissue. Got it? बाहर वाली लेयर सिर्फ क्या बनाएगी? Epidermal tissue system. Rest of the work is going to be performed by the inner part. What is the name of the inner part? Corpus. Now, here I would like to add one point, that the name of the scientist is Haberlandt. He has given the type of meristem on the basis of function. Yes, अगर मेरिस्टेम का काम है एपिडर्मिस बनाना, meristem is involved in the synthesis of epidermis. This is known as protoderm. Right? So this naming of the meristem on the basis of function, it was given by Haberlandt. If the function of the meristem is forming the ground tissue, then it is known as ground meristem. Right? And if the function is forming the vascular tissue, then it is known as procambium. The procambium can form vascular tissue, xylem phloem. Xylem phloem, along with that intravascular cambium, which is present between, which is present in the vascular bundle. Are you getting that? Procambium will form primary xylem, primary phloem, along with that intravascular cambium. So these are the three types of meristem, right? Three types of the meristem, and these three types of the meristem were the first time was distinguished by Haberlandt. Got it? Now, generally the tunica is single layer, but sometimes the tunica can be more than one layer. If the tunica is more than one layer, then it does not mean it will form multi-layered epidermis. It will be involved only in the formation of epidermis. In that case, only the outer layer forms epidermis and inner layers, they will form the part of ground tissue. Got it? Generally एक layer है तो क्या बनाएगी? Epidermis. पर एक से ज्यादा layer है अगर tunica. So only the outer part will form epidermis and inner layers, they will forming the part of ground tissue. So this is clear. The shoot apex organization. After that, now we are going to deal with root apex. So here I will be dealing with root apex. So now I will be dealing with root apex. So let's make the structure for this and then I will explain it. Yes, in the root apex, the part which I am showing here, this part, this is going to represent the root apical meristem. The part which I am representing here, this is root apical meristem. So you can easily say, see here, the root apical meristem is protected with the help of this covering. This covering protecting the meristem, representing the root cap. Due to the presence of root cap, it is subterminal in position, which we have discussed right now. Now, the cells of the root apical meristem, they have the meristematic activity. When they show the meristematic activity, they will add the cells towards the lower side, as well as they will add the cells towards the upper side. एपेक्स की तरफ भी सेल ऐड होगा और बेसल साइड की तरफ भी सेल ऐड होगा इन कंट्रास्ट टू द शूट एपिकल मेरिस्टेम, जहां पर सेल सिर्फ कहां पर ऐड हुआ था? बेसल रीजन की तरफ. इन द शूट एपिकल मेरिस्टेम जब वो ग्रो करती है, देयर इज ऑल्टरनेट ब्रॉडनिंग एंड नैरोइंग ऑफ द रीजन, बट दैट टाइप ऑफ द कंडीशन इन द रूट एपेक्स इट इज एब्सेंट. So you're getting it? Subterminal किसकी वजह से? Root cap, apical, as well as basal region, cells are cut. And now the broadening and narrowing of the apex, it is absent. So got this? So here again now we have to see the different theories which explains the organization of root apex. Different theories explaining organization of root apex. Two theories I will be dealing here. First is the histogen theory given by the Hansteen. Yes. According to the Hansteen, in the dicots, there is presence of 3 layers, 3 histogens in the root apex. The same 3 layers which we have studied earlier. Yes, please name them firstly. Dermatogen, then peribulum and plerome. I think the distinguishment of the 3 layers is very much visible here. Outermost, dermatogen, then peribulum, then it is plerome. The function of all these three layers is very well known to you now. Now, among these three histogen, which will be forming the epidermis? Epidermis will be formed by dermatogen, but along with the epidermis, one more function it will perform here, that is formation of root cap. Along with epidermis. Now, it will also be forming root cap, right? Now let's see what is going to be happening, what is going to be there in monocots. In the monocots, instead of the three, now there is presence of the 4 layers. ये 3 layers तो present ही हैं. Yes, ये तीनों तो present ही हैं. इसके साथ साथ एक और लेयर बाहर की तरफ डिस्टिंग्विश्ड होगी, that is calyptrogen. On the outer side of the dermatogen, there is formation of calyptrogen, and this will be forming root cap. Got this or not? So, dicots, the root cap, and epidermis formed by dermatogen. And in the case of the monocots, now for the formation of the root cap, one additional histogen is formed here, that is calyptrogen. Now the next theory, that is called Capet theory. This was given by the scientist Skew. Now this theory is almost similar to the tunica carpus theory. Among these 2 layers, yes, carp and cape is going to work like the tunica, means forming the epidermis. Write E for epidermis, epidermal tissue system. Now this carp, this is working like corpus. So forming ground tissue and vascular tissue. Got this? Now, after this, I would like to introduce one term here, that is quiescent center. Yes, quiescent center. Quiescent center in the root apex, it was first discovered by the Cloves in maize. Yes, according to the maize, ये जो रूट है, ये जो रूट एपेक्स है, वहां पर एक inverted cup shape structure है. There is a inverted cup shape structure. Yes, inverted cup shape structure near the apex. Yes, which I have marked here. This is known as quiescent center. Now this structure, here the cells which are present, they have low mitotic activity. Root meristem का part है, but it has low mitotic activity. DNA, RNA, protein content is less. These content, they are less. Now it means mitotic activity is less, so it means now this part is in the resting stage. Can we say low mitotic activity? So surely you can say it is in the resting stage. Due to this resting stage, it is known as quiescent center. जब तक main meristem intact है, the cells of the quiescent center, they will remain in the resting stage, right? अगर किसी रीजन की वजह से मेन मेरिस्टेम ब्रेक हो गई है, मेन मेरिस्टेम इंजर्ड हो गई है, then the function of the main meristem is performed by this center. Means, depending upon the requirement of plant, Depending upon the call of plant, it will work like मेरिस्टेम. तो नॉर्मली कौन सी स्टेज पर रहता है? रेस्टिंग स्टेज. जरूरत पड़ने पर किस में कन्वर्ट हो सकता है? मेरिस्टेम के अंदर. ड्यू टू दिस नेचर द क्रिसेंट सेंटर इट इज नोन एज रिजर्व मेरिस्टेम. रिजर्व meristem. Generally in resting stage. Depending upon the call of the plant, it can be converted into meristem. That's why it is known as reserve meristem. So this is all about the meristematic tissue. Here we have studied meristematic tissue on the basis of position, on the basis of location, as well as we have studied the shoot and root apical meristem in detail. Now, in the next class, we will be starting with the permanent tissue. Right? Thank you so much.